Hi class, and welcome to this week's video on DNA structure. We are just starting our molecular genetics unit. We're going to learn a little bit about DNA structure, its replication, and then eventually how that DNA is transformed into RNA and we use it to make proteins. So let's go ahead and get started with this. Now, I'm going to tell you about many different scientists in this unit. I would keep a running list of what they did and how they are important to DNA structure, DNA discovery, because some of these you will find in the book we are currently reading, The Double Helix, and I would like you to be able to relate back to that. So let's start with Frederick Griffith. Frederick Griffith, in 1928, he ran an experiment using 2 strains of Streptococcus pneumonia. And what he did was he had 2 strains and he treated mice with that. What we found out from this is something was changing. We just didn't know what was changing. As you can see through our picture here, that we have 2 different strains and both of them were injected. If you have the rough strain though, the mouse lived. However, when we heated and killed both the rough strain and the smooth strain, the mouse died. Now, we would have thought that the mouse would have lived, and the reason the mouse died was through DNA transformation. So we're going to go into a little bit of exactly what happened as far as the DNA. You're not going to get the whole story on the mouse. If you want to get the whole story on the mouse, you can go ahead and look it up. Now, after Griffith did his experiment, 3 more scientists were very interested in his experiment, and these 3 scientists were Avery McLeod and McCarthy. And these scientists knew that Griffith had thought there was something within the cell that it had to do with something there, but they didn't know what it was. They knew that the DNA was there, and they did additional tests on the Streptococcus pneumonia. And what they found out, that DNA was the key component of Griffith's work, and when that key component was that DNA, we knew DNA was there and it coded for our genetics. So that was a huge discovery for us. Now, after Avery, McLeod, and McCarthy, Hershey and Chase set up an experiment because we wanted to know exactly what this DNA was, what it was doing for us, because we could see that it was very, very important, but at this point we didn't know exactly what was happening. So Hershey and Chase used bacteriophages. As you can see, we have 2 different bacteriophages over here, and these bacteriophages are just a virus that infects bacteria. So they won't infect you or me, it will only infect different types of bacteria. And what he did was he labeled the capsule and he labeled the DNA. And if you know anything about the virus cycle, when it attaches to a host cell, because a virus is not a full organism, it only has a protein coat and DNA core. And when it attaches to a cell, it injects that DNA. So we could see, based on the coat being labeled and the DNA being labeled, what happened with the DNA. And when that DNA was injected, we could see exactly that it was starting to make more viruses. This was due to the coding of the DNA. So we knew, based on that the DNA is what causes a change and where we store genetic information. We were able to go up the chain from this instead of just thinking that it was the virus and the bacteria. So let's go on to a little bit of DNA structure. You should remember this from beginning of the year, but I just wanted to have you relearn it or refresh yourself on it. So DNA stands for deoxyribose nucleic acid. At the beginning of the year, we talked about all macromolecules have monomers, and the monomer of DNA is a nucleotide. Now, nucleotides, remember, are composed of a 5-carbon sugar, which in DNA is called deoxyribose. It is made of a phosphate and a nitrogenous base. Now remember, that 5-carbon sugar, that deoxyribose, and that phosphate are what make up the backbone of the DNA, and we'll talk more about that backbone of the DNA. At this point, I would like you to draw the basic structure of a nucleotide, and you have a structure, a molecular structure. I want both of those in your notes, so go ahead and take notes or take a screenshot. Now remember, we just talked about those nitrogenous bases, and the nitrogenous bases in DNA, now remember this is different than RNA. DNA and RNA are similar, but they're not exactly the same. In DNA, our bases are adenine, which is A, guanine, which is G, cytosine, which is C, and thymine, which is T. We have no U in DNA. Now, in our nitrogenous bases, we have what we call purines and pyrimidines, and that is basically just describing their structure. The structure of a purine is 2 rings. So as you can see, the adenine here has 2 rings, and the guanine has 2 rings. Their structures are a little bit different, lending to their characteristics, but pretty much you're going to see them drawn almost exactly the same, just like this. I would like you to also copy this down into your notes. Now, so we just went over purines. Purines are double ring. Pyrimidines consist of a single ring structure. As you can see, we only have 1 ring here, and they are made of many nitrogens instead of lots of carbons. Here we have cytosine, which is a pyrimidine, a single ring, and thymine, which is a single ring. So the bond, when we talk about the bonding in DNA with a double ring and a single ring, it's going to be very important when talking about pyrimidines and purines. So we'll come back to this later when we talk more about how Watson and Crick discovered the double helix. Okay, so Edwin Chargaff. I don't know if you've heard of him, but he has a law that is very important to biology, to DNA. And what he did was he discovered that when he was looking at different samples of DNA from various organisms, that the percentage of guanine, or G, and the percentage of cytosine, or C, were equal in the DNA. Now this isn't talking about DNA from a flower and DNA from a frog. This is just saying the percent of guanine and cytosine were basically the same in the DNA that we found. He wasn't comparing different species at all. And as the same, the percentage of adenine and the percentage of thymine are equal in DNA. So what he found was A will bond with T and C will bond with G. Now that was very interesting because you have a single bond, or a single ring, and a double ring being bonded together. Let's take a look at our DNA structure then. So DNA structure, like we've talked about, has a 5-carbon sugar and phosphate, and that makes up our backbone. As you can see right here, the sugar phosphate backbone. This is going to be your phosphate. You see the P and 4O, so it's got a molecular formula of PO4, and then you see the 5-carbon sugar. You can count the ends here around the sugar, and that's going to be your deoxyribose. Now, when you look at our nitrogenous base, we have adenine in green, thymine in pink, and you can see these bonds right here. These bonds are called hydrogen bonds. I know we talked about that earlier in the year. These hydrogen bonds are very important to us, and this is what holds basically that double helix together. Now, in adenine and thymine, you have 2 hydrogen bonds, and in cytosine and guanine, you have 3 hydrogen bonds. The easiest way I can tell you to remember this is A and T spell a 2-letter word, which is at. So the 2-letter word has 2 bonds, and the other one has 3 bonds. Okay, that looks like that is all for today. Go ahead and copy all these figures into your notes, and we will talk about this later in class.